Hello everyone, welcome to my this video in YouTube channel. Through this video today I'm going to let you know about biological control made simple, which is also I have given title as Antibiosis versus Plant Immunity. So, did you know some bacteria don't kill pathogen but still protect the plants? How? Today we will learn about how bacillus protect uh, plants from fusarium species of fungi with a simple hands-on understanding. Okay, so fusarium oxysporium is a fungus which is harmful to plant, causes plant disease, and bacillus is a beneficial bacteria we can get on soil. So bacteria can help a plant in two ways, like antibiosis, they kill the fungus directly, or they activate the plant immune system. In both of the way they can support mm, for the plant health. Not all bacteria are good at killing fungi. Yeah, so some train plant to defend themselves as well. So to do this experiment to justify, I'd like to let you know about step by step methodology how to conduct this experiment. So initially we have to collect the sample from root from healthy plants. After having samples from on the rhizospheric area, we have to isolate a bacteria on a nutrient plots, like we can initially start with the nutrient agar plate and select different colony from there. Then we put bacteria on other fungi cultivated media on the same state, like we put the bacteria bacillus near to the fungi cultivated in potato dextrose agar, and we look for the zone. If we find a clear zone, it means definitely bacteria is inhibiting fungi, so that could be antifungal. But if the case is uh, there is no zone of inhibition around plate of fungi cultivation, then that would be non-antifungal. So scientists found what there is no inhibition but still work later. So hands-on simulation includes different ideas. Okay, at a beginner level, what we will do, we have a fruit or bread which is uh, infected or affected by the fungi, or even we can take for a bacteria as soil bacteria or yeast. So for a real plant experiment, what we will do, for real plant experiment, we grow plants in the control environment and add bacteria. Then we infect with the fungi as well and look for the results. If there is a impact of bacteria, uh, if they kill, it means definitely they protected plants. If they did not kill, it means we have to understand they are boosting their immune systems, stimulate. So is they activate the plant's defense systems, like a salicylic pathway, jasmonic acid pathway after the infection, and they function as a priming, like a stimulator. So they have faster and strong response when they attacked with a disease. This is called also induced systematic resistance. In conclusion, we have two strategies to protect our plants. One is to kill a pathogen through antibiosis, like using a bacteria, and boost the plant immunity with the same priming process. Why it is important? Because they are not chemicals. It means we will have a utilization of less chemicals, sustainable agriculture, better long-term productions, and the best bacteria don't just fight, but they also teach a plant how to fight and how to boost. Thank you so much. Please like, share, and subscribe my this video for a better update and for coming new videos.